Hello guys, it's Ivan, and here I am joined with Mario Medelis.
Hello guys, I'm Mario.
Today we are going to talk about genetics and genetic engineering. Now the main thing we are supposed to answer is why genetic engineering should be allowed on organisms, specifically towards medical research.
First, let's touch down on genetics. In every cell there is DNA. DNA is a nucleic acid that contains information for the cell. Cells need this information to produce any necessities. In DNA there are nucleotides. These nucleotides are adenine, thymine, guanine, and cytosine. These nucleotides contain the actual information for DNA. Now the rule for DNA is that adenine will always bond with thymine, and guanine will always bind with cytosine, and vice versa.
These bonds are strong and can only be broken by an enzyme, DNA helicase. This enzyme helps to unzip the genes. The only other thing that can break the bonds of DNA is mutations, which we'll talk about later. DNA is only unzipped to produce a duplicate strand or to create mRNA. This process is called DNA replication. In this process, DNA is unzipped. When DNA is unzipped, nucleotides are floating around, bond with both sides of DNA. This causes two strands of DNA to be made, one old and one new. Because of this, DNA is considered semi-conservative molecule, meaning that half of the DNA is old and half of the DNA is new. When stretched out, the DNA of one cell is 2 meters long. How does it manage to fit in something so small even though it's that big? More specifically, how is it supposed to give information to the cell? The nucleus seems too small to fit all that DNA in, so how is it supposed to send out all that information to the rest of the cell if it can only stay there? Well, the solution to this is mRNA. mRNA is another nucleic acid. Unlike DNA, it only contains one gene. This is perfect because the outer part of the nucleus has openings for only the tiniest molecules to pass through. And since it only contains one gene, it can fit through the nucleus to get to other parts of the cell. Another difference between mRNA and DNA is that mRNA contains uracil in place of thymine. It will still contain the same genetic message with uracil, but uracil is more primitive than thymine. Thymine is evolved to be more genetically stable and to not be messed up as easily, but uracil is easier to transfer. mRNA is also single-stranded to suit the size of nuclear pores. Well, mRNA is different than all the other ones because it can send messages from the nucleus. The mRNA simply gathers the message from the nucleus. These messages can go from fixing a part of the cell or to just build a part of the cell altogether.
But there are a lot of RNAs like messenger, transfer, and ribosomal. What does mRNA do that the others can't?
Well, mRNA is different than all the other ones because it can send messages from the nucleus. The mRNA simply gathers the message from the nucleus. These messages can go from fixing a part of the cell or to just build a part of the cell altogether.
So is that how amino acids are made?
Why, yes, dear boy.
Oh damn. The following process is translation. This requires an enzyme to change any nucleotides to amino acids. To create a protein, the enzyme must first receive the start codon, and to end it, it must receive a stop codon.
Okay. Well, what would happen if it never receives a stop codon?
Well, then the protein would never officially be made. It wouldn't be finished.
Wouldn't it just continue if there isn't a stop codon?
That would be the case if the enzyme continues to receive a message from the nucleus.
What would happen if the message is altered or messed up?
Glad you asked. That would be called a mutation.
Ah, so like the X-Men, right?
Well, yes, but no.
Wait, what?
I mean, it changes people, but not to such an extreme like X-Men.
Ah, okay. So what are some mutations?
Well, a good example is heterochromia. It is a mutation in which a person has a different eye color in both eyes.
I've seen dogs with different eye colors.
Why, yes. Mutations don't only happen in humans. They happen in all organisms.
Wow. So like two-headed animals?
Those are mutants, but how does that work? Okay, Google. How does a mutation happen?
You don't need to Google that. I can explain. Now, mutations occur when DNA goes wrong, and there are many ways it can go wrong. Now, for a mutation to happen, something must go wrong, obviously. All mutations occur with either exogenous or endogenous factors.
What does that mean?
Endogenous are factors that can happen inside the cell. Now, exogenous can occur by radiation, sunlight, smoking, or anything that happens to you that are not from the inside. Endogenous factors are simple mistakes made or their errors caused by toxic byproducts from the cell. Now there are two main kinds of mutations, point mutations and frameshift mutations. Frameshift is broken into both addition and deletion mutations. Now point mutation is when only one nucleotide is changed to a different letter.
Like if you had AUG, a codon that starts the protein to be made, and it was changed to ACG. That's a point mutation, right?
Correct. Point mutations hardly ever change the outcome of a protein. However, frameshift mutation can change the whole gene. In DNA, there are reading frames. These are in groups of 3. In a frameshift mutation, the frames will stay in the same place, but because of a base added or deleted, it can mess up everything. Like if you had UGG, CGC, AUU, if you deleted the last G in UGG, you'd get UGC, GCA, UU.
Wait, but there's not a third nucleotide. What would happen if there's only 2 nucleotides?
Then the amino acid won't be made. Alright then, take this example. If I had a sentence, the cat ate the fat rat, and deleted one letter, the entire sentence would shift to the left side. So if I remove the T in cat, it wouldn't be the cat ate the fat rat. It would be the cat tet hef atter at. How am I supposed to read this? See how it doesn't make any sense? Now, would it be possible for us to make our own mutations?
It is possible. Well, kind of. A mutation is a permanent change in a DNA sequence. There is something called genetic engineering in which we can artificially change the sequence of DNA to give organisms and plant-specific traits. So in a way, it can be thought of as an artificial mutation.
So, like giving pigs wings and stuff like that?
The process varies depending on what is being worked on. Scientists use bacteria for cures and to genetically engineer plants. Now to do this, scientists mess around with bacteria, specifically their plasmids, and plasmids that are small rings that are empty. Now these allow foreign DNA to be inserted. By doing this, it permanently changes the DNA of the bacteria and causes it to be passed on to its offspring. Scientists use this method to create things like insulin, and it is also used to genetically engineer on plants. On plants though, the bacteria infects the plant. Infecting the plant causes the DNA of the bacteria to become a part of the plant's DNA.
Well, aren't we supposed to be focusing on research on organisms? And genetic engineering is different in animals. To modify an animal's genes, scientists must get a fertilized egg of an organism, then add the exogenous DNA to the egg. This doesn't always work though. The success rate is low. If it does work, then the offspring will have the decided traits and the future generations will carry this alteration.
Well, why do we do this? Is it just a mad scientist thing?
The main reason is for research, more precisely, medical research. For example, scientists in China have created genetically modified monkeys that are born with deadly diseases like Parkinson's, Alzheimer, and other diseases.
Aw, poor monkeys. Well, this is for research, so sit down and...
You might want to stop there. This is to find a cure for diseases.
That's good, but isn't it immoral to mess with nature? And what if a mistake happens? What would you do? No, just stop. Genetic engineering succeeding is rare, but the good that can come out of it has unlimited potential. I don't believe you, it's just unethical.
Don't believe me, just watch. Don't believe me, just watch.
See? Genetic engineering actually has many solutions. Like those bananas, I prefer to eat a banana rather than get a shot any day. And we could treat cancer with eggs.
Eggs! Well, we don't know how it will work. No, I don't want genetic engineering.
Well, we cannot advance in genetic engineering if we don't try it.
Oh, I see.
Genetic engineering can also be done on humans.
What? No, that's too far.
Calm down. If we were to genetically engineer on humans, it is possible that we can cure any genetic disorders and diseases. For example, if diabetes is genetic in your family, with genetic engineering you can prevent your future offspring from the risk of diabetes.
Or if you simply want your baby to have different traits like blonde hair, colored eyes, but that's kind of too advanced for us.
Well, let's focus on the medical advantages, okay, buddy?
Okay.
Think about it. If we were to genetically engineer on humans to cure diseases, so much suffering can be prevented. We just have to take the leap of faith for research. If it is irreversible, would it really matter if it is improving human condition?
True. Why would you want to reverse a cure?
Thank you for your time, and please consider the information given. Well, how about this one?
No, it doesn't seem that good.
Well, we're gonna put it in, bro. It seems
No,
perfect.
no.
We're gonna put it in.
It doesn't.
It
look
seems perfect. It's
decent.
perfect.
No,
It's just perfect
no,
one.
no.
It's so beautiful.
No,
Look
well,
at it. Look.
Like, it doesn't even look that good.
It looks amazing. Just look at it
No,
from
I
a
don't-
scientist's perspective.
No, well, the scientist's
Mr.
perspective.
Byersdorf would approve.
The scientist would say no. That's bad.
Byersdorf would say yes.
No, it's just really, really dumb.
Inspirational.